Hello everyone. This one for ENES120 class on Monday, November 21st. And so at first, let me share my PowerPoint file. Okay. ENES120 Biology for Engineers. And summary of previous lecture is the cell. As we know, cell is composed of mainly three parts, and the cell membrane, nucleus, and cytoplasm, as you know, and the cell and the eukaryotic cells is plant and animal cells with a nucleus and membrane enclosed organelles. And the prokaryotic cells is unicellular organisms without a nucleus or membrane enclosed organelles. So this cell contains many organisms, and including mitochondrium is respiration happens, and also energy factory, and nucleus and the DNA occurs, and Golgi apparatus and cell membrane, endoplasmic reticulum, ribosome, and cytoplasm, and so on. So is DNA, as we know, is a cell. Inside the cell, it has nucleus and the chromosome. And the chromosome is composed of nucleosome. It can be divided into DNA and histone, and the gene, nucleotide, and the kind of things. And the protein is combination of amino acid, and amino acid is composed of alpha carbon atom and the amino group and the carboxyl group, and the hydrogen molecule is attached, and the side chain. So it's a cell membrane is basically fluid mosaic model is most commonly used, and it composed of phospholipid bilayer and the hydrophilic head and the hydrophobic tails, and it's protein, many proteins, protein channels, and cholesterol and glycolipid. You can see this one. So, and the cell membrane is basically selective permeability, and the cell membrane is fluid mosaic model and phospholipid protein and the kind of things. So it's small and uncharged molecules like oxygen or carbon dioxide, and this kind of molecules can easily move through the cell membrane. And okay, so however, other things like sodium and potassium, we can use large and charged molecules like sodium and potassium. You can active transport with spending energies. ATP becomes ADP, and then it's sodium and potassium can transfer. So cell wall, and this is the previous related lectures. And then chapter 12, transport across cell membrane. So the first chapter contains principles of transmembrane transport. And the transporters and their functions, and the ion channels, and the membrane potential, ion channel, and nerve cell signaling. So at first, the transmembrane transport, cell membrane transport, and this one is we will do this again on next lecture, not for today's lecture. And basically, cell membrane transport can be divided into two parts. One is passive, another one is active. So it's and the passive cell membrane transport is basically diffusion, and the diffusion and the facilitated diffusion is naturally occurring without any, doesn't need any energy. So it's like a diffusion, oxygen and carbon dioxide, osmosis, hypotonic, and isotonic. And facilitated diffusion is glucose transport. And this one is all passive transport. And active transport is we use ATP as a energy source. So with spending energy and protein and ATP and sodium and potassium pump and endocytosis, exocytosis. We will study in detail later. So it's like osmosis is diffusion of water through the semi-permeable membrane. That's the osmosis. And it can be divided into three parts. One is hypotonic, another one is isotonic, and hypotonic. Hypo means under one. So hypo means excessive usually. So it's hypotonic is high concentration. So it's water molecules move out. Isotonic is equilibrium state, and hypotonic is under concentration. So water molecule can come in. So that's the three state, and the extracellular space, and the sodium ion and the potassium ion can move through with spending ATP, and ATP becomes ADP, and with this one, sodium and potassium ions can move. And endocytosis is moving cells inside one is endocytosis, and exocytosis is release the signal molecules, and that's the exocytosis one. And the membrane transport is plasma membrane and regulate movement into and out of a cell. And this one is protein and the phospholipid one, and intestinal fluid outside of cell, and the cytosol, inner part of cells. And this is facilitated diffusion can be divided into two parts. One is channel mediated, another one is carrier mediated. The difference is type of protein used. And the channel mediated one is ions or small molecules move through a water-filled channel and stay channel mediated. And the carrier mediated movement of small polar molecules across the membrane. That's the carrier-mediated osmosis. So, and the active process require cellular energy, two types, active transport and vascular transport. And they use ATP as an energy source. That's the one. Active transport, movement of solute against the concentration gradient. And that's the vascular transport is transported by vascular membranous sac. So active process is active transport. Primary active transport is ion pump. So this is the one. So cell membrane contain specialized membrane transport protein, like this one. And so it's a protein-free artificial lipid bilayer. We call it liposome. It's only specific molecules can move through this one. However, it's a cell membrane which containing proteins is membrane transport proteins. Using this one, other molecules also can move through the membrane. So lipid bilayers are impermeable to ions and most uncharged polar molecules. The ion concentrations inside the cell are very different from those outside. And differences in the concentration of inorganic ions across cell membrane create a membrane potential. Cells contain two classes of membrane transport proteins, transporters and channels. Solute cross membranes by either passive or active transport. And both the concentration gradient and membrane potential influence the passive transport of charged solute. One is concentration gradient, another one is membrane potential influence. Membrane potential, and this is the two main things that influence the passive transport of charged solute. So this one is important. So water moves across cell membranes down its concentration gradient, a process called osmosis. And the rate, if we see, is different molecules, the small non-polar molecules like oxygen and carbon dioxide and this kind of things is artificial lipid bilayer. It can move through the layer almost most of these molecules can move through. However, it's a small uncharged polar molecules like water and ethanol and the glycerol is most of it cannot move through. However, still some part is okay. However, larger uncharged polar molecules like amino acid and glucose and nucleoside, and this one is much less portion of molecules can move through. However, it's charged ions, hydrogen, sodium, potassium, calcium, and this kind of things, it cannot move through at all. So the artificial lipid bilayer. And then how can these molecules can move through this using active transport through the protein. So it's like a comparison of ion concentration inside and outside a typical mammalian cells, and the sodium, potassium, and the magnesium and calcium, and this one is almost 10 times difference, right? At least 10 times and 30 times difference, and dozen time difference. Also, this one is similar, you can see. And in organic ions and small polar organic molecules can cross a cell membrane through either a transporter or a channel, and the channel is ions you can move through using this cell membrane. Another one is transporter. It has solute binding site, and it moves like this one. So another one is solute cross cell membranes by either passive or active transport. It's a cell membrane. It's simple diffusion one. It's also possible. However, bigger one, you can move through using channel mediated or transporter mediated or active transport with spending energy and pump one. And the concentration gradient is like this direction, assuming concentration gradient is this direction. Actually, this is the summary of these chapters. So electrochemical gradient has two components. One is when voltage and concentration gradient work in the same direction. Voltage and the positively charged one, and here negatively charged one. In that case, positively charged ions can move through this way by voltage gradient. Another one is concentration. It's higher concentration, is lower concentration. So concentration gradient is this way. In this case, it's same directions. So it's like a strong force. Many molecules can move. However, other case, like voltage gradient is like a positive to negative this direction. However, concentration gradient is like this direction, so opposite direction one. In that case, work in opposite directions. And this one is water molecules diffuse rapidly through aquaporin, these channels in the plasma membrane of some cells. There's water molecule and membrane. So cells use different tactics to avoid osmotic swelling, and protozoan it has eject water, and plant cells it has cell wall. Cell wall is relatively solid one, so it can avoid swelling, osmotic swelling. Animal cell case, we use ion pump to avoid osmotic swelling. And the passive transporters move a solute along its electrochemical gradient. Pumps actively transport the solute against its electrochemical gradient. And sodium pump in animal cells use energy supplied by ATP to expel sodium ion and bring in potassium ion. Sodium pump generate a steep concentration gradient of sodium ion across the plasma membrane. And the calcium pump kept the cytosolic calcium ion concentration low. And the gradient-driven pump exploit solute gradient to mediate active transport. And electrochemical sodium gradient drive the transport of glucose across the plasma membrane of animal cells. Electrochemical hydrogen ion gradient drive the transport of solute in plant, fungi, and bacteria. So each cell membrane has its own characteristic set of transporters and nucleotide and sugar, amino acid, and the sodium potassium pump, and ribosome case hydrogen molecule transport. And mitochondrion one is ATP binding pyruvate, and this one. Conformational changes, transporter mediate the passive transport of solute. And it's like a glucose case is glucose transporter when the glucose is combined, and then it changes the shapes like this way. And then release the glucose, transport glucose through the cell membrane. So gradient-driven pump is like this one is concentration gradient is this directions from upper part to lower part, and then is combined with this one, and then this case this molecule moves this way. So gradient-driven pump is this one. ATP-driven pump, ATP combined, and then energy spending, and then move this one. Another one is light-driven pump. When light is injected and then activate this protein and then transport these molecules. And the sodium pump use the energy ATP. It says sodium electrochemical gradient is from this to this directions. And then how can we transport this in opposite direction one with spending energy, ATP. So that's the one. Sodium pump undergoes a series of conformal changes as it exchanges sodium ions for potassium. At first, the sodium ion bind and pump phosphorylate itself, hydrolyzing ATP combined, and then transport sodium. And then it combined with potassium ion bind, and then this phosphorylate is released and the pump, and then move this pump return to original conformation, and potassium ion is ejected, and this one. At first, sodium ion and then potassium ion combined, and then release this one. So how about calcium? And the calcium pump is similar one, using spending ATP and ADP, and then transport. through the cell membrane. And gradient-driven pump can act as sym port or antiport. Okay, sym port is like in this case, transported molecules and co-transported ion. It's the same direction, and that's the sym port. If it's opposite direction, as anti-transported ion is like this direction, and the transported molecule is opposite direction case, and then we call it antiport. And only one transported molecules move case is, we call it uniport. So extracellular space and glucose sodium sym port use the electrochemical gradient, electrochemical sodium ion gradient, in this case, this direction, to derive the active import of glucose. So at first, sodium ion combined, and then glucose also combined, and then release like this way. So in that case, glucose gradient is opposite direction one. However, sodium gradient is this direction. So when sodium ion move through these directions, and glucose also combined. So that's the one. And the two types of glucose transporters. One is sodium-driven glucose sym port. One is this one. Another is passive glucose uniport, and the passive glucose uniport. In this case, sodium glucose combined move through this one. However, this passive glucose uniport, only glucose molecules can move through this one. So that's two kind of glucose transporters. Animal cells and plant cells use a variety of membrane pump. That's like a sodium pump and the sodium-driven sym port, and this one, hydrogen pump, and this kind of things is used. However, animal cells, it's a hydrogen pump, hydrogen-driven sym port. And so different mechanisms are used in animal and plant cells. And this is some examples of transmembrane pump. So sodium-driven glucose pump, and sodium hydrogen exchange, and sodium pump, sodium potassium ATPase. Excuse me. And the calcium pump, and so on. Ion channels and the membrane potential. Ion channels are ion selective and gated, and the membrane potential is governed by the permeability of a membrane to specific ions. Ion channels randomly snap between open and closed state, and the different type of stimuli influence the opening and closing of ion channels. Voltage-gated ion channels respond to the membrane potential. So an ion channel has selective filters here. In this case, is narrower. So at first, water molecule is combined, and the potassium ion at first combined with water molecules. However, in this stage, it cannot move through this way. Only potassium ion can move through this way. So this is the filter, selective filter 1. Ion channel has a selectivity filter. So ion channel fluctuate between closed and open conformation. This one is closed case, and the selectivity filter is open state. And like this is the Venus fly trap, use electrical signaling to capture its prey. So this is the example of that one. And distribution of ions on either side of cell membrane give rise to its membrane potential. In this case, it's equilibrium states. However, this one is more positively charged here and the negatively charged this one. So it's like a potential difference. And extracellular space in here is concentration difference is happened, and also in this case is positively charged and this one is negatively charged. So voltage difference, in other words, potential difference. And potassium leak channels opens. In this case, is leak channel opens, and the membrane potential exactly balance the tendency of potassium to leave. In this case, it's leave. And so it's a Nernst equation. Nernst equation is force tending to drive ion across a membrane is made up of two components. One is electrical membrane potential difference, and another one is concentration gradient difference of the ions. Equilibrium states it has voltage, has concentration outside and inside the concentration, and V is the membrane potential case. It's Nernst equation, and this one is important. So you need to memorize. And the patch clamp recording is used to monitor ion channel activities. So it's at first we remove this ion channel, some part of ion channels, and then eject recording microelectrode, and then measure the electrical potential, electrical voltage difference, and then recording one like this. And the behavior of a single ion channel can be observed using like open, closed, open, and closed. And the potential difference is current difference. So a case is a neurotransmitter acetylcholine is present case, and then it's we can measure the voltage difference. In B case, the neurotransmitter is not present case. We cannot see any potential difference. So different types of gated ion channels respond to different type of stimuli. So in this case, voltage gated. And here, positively charged and the negatively charged one, and then it can be activated. So this is the voltage gated. Another one is ligand gated. Ligand combined, and then it is opened. Ligand gated, extracellular ligand. And this one is outside ligand, and this one is inside intracellular ligand gated case, and this one is mechanically gated, mechanical force. So example of mechanically gated ion channels is our hearing part inside ear. So stereocilia, and this one is mechanically gated ion channels. And this one, when mechanically is tilted, and then this channels opened around. So both mechanically gated and voltage gated ion channels underlie the leaf closing response. Leaf is closing one, and this one is mechanically gated and voltage gated, and both of them are involved. So this is end of class for November 21st one. And if you have any questions, please email me. Okay. And thank you and bye.